All right, so last time, how many of you were at the last one of these? Hold up your hand. So most of you. We talked last time about taxonomy, which is the system of like classifying things into different groups. And we talked about how that the system that we're using today is phylogenetically based, which means it's based on evolution and genetics. When two things are in the same group, it means they're related through genetics and evolution. All the different subgroups are called clades, and we've got several of those. The biggest clade is the domain. There's three main domains. Eukarya, which includes all the eukaryotic organisms like plants and animals, protists and fungi. Eubacteria, and that includes what we would call normal bacteria, normal prokaryotic cells. And then the archaea, which used to be called Archaeobacteria. But what scientists have discovered is that they're genetically somewhere in between these two. They're more closely related to eukaryotes, possibly, than they are to other bacteria. And also, eukaryotes are more closely related to archaea than to eubacteria. The next group down is the kingdom. And depending on which textbook you look in, you may see 5 kingdoms, you may see 6, you may see 7. We're talking about things like the animal kingdom, the plant kingdom, the fungi kingdom, and so on. Phylum is the next smaller group, and we just work our way down. As you go down, the groups are less inclusive, meaning there's less things in there. And if two things share the same family, what does that probably mean about those two things? They're really closely related. If they share a genus, they're even more closely related. If they share the same species, they are the same thing, right? So that's the way the system is set up. Remember prokaryotic versus eukaryotic. If an organism is prokaryotic, what's it lacking? Yeah, let's be specific with our terminology. Prokaryotic cells lack, let's just don't say a nucleus, let's say a membrane enclosed nucleus. Meaning that their DNA isn't separated from the rest of the cell. It's just kind of floating out there, right? They also lack what else? Yeah, membrane-bound, what? Organelles. Like mitochondria, chloroplasts, and such. We talked about how those things are really, really important in eukaryotic organisms for dividing up the work and what? Yeah, but with a bacteria, that one cell has to be able to do everything. So it's really not very good at what? Anything. But in a eukaryotic cell, each part has a certain way, certain job. So the parts become specialized, and they become way, way more what? Efficient, better at doing their work. So on Earth today, the only things that we might characterize as prokaryotic would be archaea and eubacteria. Every other living thing on Earth is a eukaryote. Every other thing on Earth has membrane-bound or membrane-enclosed nuclei and membrane-bound organelles. This thing is a phylogenetic tree. That's not where I wanted to go. Let's try that again. And what a phylogenetic tree is supposed to show you is sort of like an evolutionary family tree. And remember, down at the base of this phylogenetic tree, what's implied to be there? The first life forms, which in that Edmodo assignment they called what? Protobionts. Yep. Bacteria like things. And then right up here at this V in the line, that's implied to be a common ancestor between what? Whatever branched off this way and whatever branched off this way. So it's the common ancestor between the eubacteria branch and the everything else branch. And then right in here, that's supposed to be the common ancestor between what? The archaea branch and the eukarya branch. Remember, the lengths of these lines loosely are supposed to represent what? Time. So that you can see that the archaea, in terms of time, evolved more closely to eukaryotes than they did to than eubacteria did to eukaryotes. And also, we share a common ancestor with archaea that's more closely related to us than the one we share with bacteria. We'll talk about some more with that in a minute. It's not cooperating. All right, we mentioned the kingdoms already. That in most textbooks you're going to see a kingdom for archaea, a kingdom for eubacteria. You're going to see plant, fungi, and animal. And depending on the textbook, you may or may not see Contista given their own kingdom. Because protists are really, really different kinds of things. There's the animal-like protist, which we call what? Amoebas and parameciums. What are they called? Protozoans. And then there's the plant-like protists that are called algae. And they're not very much alike. So a lot of times they're not categorized in a kingdom anymore. Because remember, if you're in the same kingdom, what's that supposed to mean? You share an ancestor, you're evolutionarily fairly closely related. And we want to go through and just remind you of where we belong in this system. What domain do we belong to? Eukarya. Kingdom is pretty obvious. Mammalia. What phylum do we belong to as humans? Chordata, which includes what? All the vertebrates. So our kingdom includes all animals. Our phylum includes only the vertebrates. What about our class? Mammalia. So our class includes only vertebrate animals that do what? Breastfeed. And then our order, what's our order? Primates. So these are only mammals with what? Opposable thumbs and well-developed brains, like monkeys and orangutans and chimps and apes and everything in between, lemurs. What about our family? Hominidae or hominid. And these are only primates that can do what? Walk upright, like the great apes, the orangutans, the gorillas, us. And then our genus is Homo. And those are only hominids, which we would today classify as what? Men. Now, are there any other living people other than us? No. But scientists would say what? There used to be Neanderthals and all those different cavemen ancestry kinds of things. And our species is sapiens. And remember, if you're going to give something a scientific name, it's always a combination of genus and species. Genus is written first and in capital letters. Species is written as like your last name, but it's all in lowercase. And when you handwrite it, you underline it. Or if you print it, what are you supposed to do? Italicize. All right, where we left off last time, we just barely started talking about bacteria. And we're going to try to talk about eubacteria today, which are your normal, old, regular bacteria. Remember, what does the root EU mean? True. These are your true bacteria. We're just going to go through some characteristics of them here first. They're prokaryotic. Again, they lack a membrane-enclosed nucleus. They lack all those organelles. They do have DNA, though. Their DNA, their genetic material, is often called the nucleoid. If you add OID on the word, on the end of a word, what does it mean? Like nucleus. How much DNA do they have? How much DNA do you have? How many chromosomes do you have per cell? You have 46. And yours are said to be, eukaryotic organisms' chromosomes are said to be linear. And we usually have a lot of them. But they're not really straight lines, but they're not what? What's the shape of a bacterial chromosome? It's a circle. It may look like that. It's a loop. Ours aren't. Ours aren't loops. Bacterial cells typically have one circular chromosome. It just sort of floats around. It's really small. They don't have a lot of DNA. They can't do a lot of the things you can do. But realize they do have some genetic material. It's just floating. It's usually one single circular chromosome. Also, eubacteria have cell wall made out of peptidoglycan. And if you look at that word from the stuff we've been talking about in class, what does it mean? Part what and part what? Part protein, part sugar. Different than a plant cell wall. What's a plant cell wall made out of? Cellulose. But regardless, the function is still sort of the same. What's the function of a cell wall? You give it some support, some structure. But in bacteria, what does it help them not do? Water can go straight through it. But there's a cell membrane underneath the wall, right? And if there was no cell wall there, let's say that Floyd here was a cell, a bacterial cell, no cell wall, and we just threw him into water. What would happen? He would start to swell, right? Because he's got salts and sugars inside of him. He'd just swell and swell and swell and swell and swell until eventually what would happen? He'd just pop. If you're a bacterial cell, you don't want to pop. So what do you build around yourself? A wall. And that wall will allow you to swell up to a certain extent, and then you can't go any further. So it helps to keep them from popping when they're in a watery solution. That's pretty important. Now, we just got through saying that bacteria don't have organelles. Every cell, though, has structures called ribosomes. They're not made of membranes. They're sort of these solid pieces of machinery. And for those of you who are in class today, what are ribosomes all about? They make the proteins based on the directions of what? DNA. Every cell that's alive has to have ribosomes because most of what we do is made to happen because of these proteins. Some bacteria have what's called a glycocalyx or a capsule. And this is sort of like a mucousy-like coat outside the wall. Why would it make sense if you were a bacteria to cover your outsides in mucus? From what? If you're a bacteria, what are your two worst enemies? Not usually. One of them is drying out. So if you put this mucousy coat on you, it helps to keep it from drying out. Keeps water in. What's a fancy word for drying out? Because you know in science we have to use big fancy words. Well, dehydration, that's not the word I'm looking for. DSOA. Maybe you don't know this word. Desiccation. If you ever see that word, and you will in this class, it just means drying out. So the mucousy capsule helps them to prevent being dried out. That's number one enemy. What's another enemy? Maybe. I don't think this would help with that, though. Immune systems. The things they're living in. Helps to protect them against immune systems. Attackers. White blood cells. Antibodies. Those kinds of things. Something I think we talked about in the last one of these sessions is that bacteria also have these little hair-like extensions called pili. And their most common job is to act sort of like bacterial Velcro, to help them stick to surfaces. But there's a few of those that are specialized. Most bacteria are reproduced through what method? Asexually, it's called binary fission, where essentially they just copy their chromosome and then split in half. What's the advantage of reproducing asexually? The advantage. Fast, and you don't have to spend energy and resources trying to attract a mate, right? That can be expensive. But the downside of asexual reproduction is what? There's no genetic variation. So let's say there was some kind of illness that bacteria could get, and they can get virus infections. Bacteria can get viruses. And let's say that one of those bacteria was susceptible to that virus. What does that mean about all the bacteria? They're all susceptible because they're all genetically identical. They'll be clones of each other. Variation helps us to avoid that. If there hadn't been variation in the human population when the Black Death came along, what probably would have happened to us? We might have all went extinct. But some of us were immune, or at least highly resistant to it. And we made it. And what does that mean about those of us still alive today? We may not be immune, but we're probably more resistant to it than the people that were alive back then, right? Because most of their ancestors died. So where I'm going is that bacteria need a method to approach something like sexual reproduction. And that method is called conjugation. And what two bacteria will sometimes do is they'll join together with these specialized phyla called sex phyla. And essentially do what? They'll swap some DNA. We just got through saying a minute ago that they only have one main chromosome. It's kind of a loop. But in addition to that main chromosome, they may have some really, really tiny circles of DNA with just a few genes on them. And those are the pieces of DNA that they will swap. And those tiny loops of DNA that they can switch, they're called plasmids. So two bacteria hook together, they conjugate, they swap some plasmids, they separate. That's called sexual reproduction. You started with two bacteria, you still just got two. So why is it called sexual reproduction? The two you started with are different than what? The two you ended with. It created variation. That's really what sex is all about. Mixing up genes, creating new genetic combinations. We mentioned prokaryotic or binary fission a minute ago. That's just another name for it, prokaryotic fission. It's the main asexual way bacteria reproduce. We also mentioned last time, I think, the different shapes that bacteria come in. There's three main ones. Anybody know one? Okay, I heard somebody say coccus. The coccus just means sphere shaped. All right, what's another one? Bacillus. And that means like shaped like a rod, like a three-dimensional cylinder, sort of. And then the other one is just kind of a little spiral shape, and it's called spirillum. Another thing I will mention is that some bacteria make these structures called endospores. Let's say that this is a bacterial cell. And here's the chromosome. And let's say something bad is happening to the bacteria. Maybe it's getting really dry or really hot. There's no food. To keep from dying, what will that bacterial cell do sometimes? When the environment starts to go really, really bad, it'll build a thick protective coat just around its chromosome. What happens to the rest of the cell? It dies. That structure is called an endospore. So it's essentially the chromosome with a highly thick protective coat around it. When the environment gets better, what happens to that coat? It dissolves, and then the chromosome will give rise to a new cell. Now that should be kind of scary to you, because some of the ways we try to sanitize things to kill germs, when we start to try to kill them, what do? They build those endospores. And if we're not careful, if we don't treat them with the right stuff or heat things up high enough, we think we killed them when in reality, we didn't. Okay? And they just grow back, and that can make us sick. Another thing that I want to talk about and try to break some myths about is a lot of people, they hear the word bacteria and they think bad. It's going to make you sick. And there are lots of bacterial diseases. Here's a short list. Strep throat, tetanus. Is tetanus better known to some people else? You all have a vaccine against tetanus. What's it about? No? It's called lockjaw. How do you usually get tetanus? You usually get it comes from these bacteria that cause this live in dirty, rusty places, typically. So when do you go get a tetanus shot? Cut yourself on something rusty, like an old nail or something like that. It's not the rust that gives you tetanus. It's the bacteria that live there. Syphilis, kind of STD, pneumonia, and there are lots of others. Bacteria. What's probably the bacterial disease that's killed more people than any one? I think of two. I may be wrong here, but tuberculosis is bacterial, TB, and the Black Death was bacterial as well, plague. You don't hear a whole lot about bacterial diseases anymore. Why not? They're treatable with antibiotics, at least most of them are. And most bacterial diseases, if you take antibiotics, you'll typically get over it. There's some symptoms. I mean, there's some exceptions. There are some bacteria that are becoming resistant to antibiotics, and that's a whole different problem. But most bacterial diseases we can now treat pretty well. What I want to fix you in on, though, is that most bacteria are actually really positive. Even though there are a few that will make you sick, most are really, really important and good. And they do things like decomposition. If there weren't things like bacteria and fungi out there, the leaves are all about to start falling off trees, what would happen in a few years? We'd be having to push our way through leaves because they just stay there. And not only is getting rid of them important, what else do the bacteria do as they break them down? All the minerals like phosphorus and nitrogen and any other mineral that's in that leaf, that ends up back in the soil so that things can use it. So sometimes we say they're mineral recyclers. They help to recycle the minerals through the food chain. We talked about nitrogen fixation, how that bacteria in the soils, in the oceans, or plant roots sometimes, will take that into that's triple binded and help to convert it into either ammonium ions or nitrate ions, which plants and animals can use. And that's important. And there are other positive roles as well. Those are probably the main ones. Another thing that I don't think we mentioned in here before is let's talk about these three terms. If something's called an aerobe, what does that mean? It uses oxygen to do aerobic cellular respiration. Some bacteria are aerobes, like you. They have to have oxygen to do respiration, which means they have to have oxygen to make what? ATP. Gives them lets them take food energy and convert it to a usable form of energy. Some bacteria have to have oxygen. Some bacteria are called obligate anaerobes. And if you look at that word, obligate, looks a lot like what? Obligated, obligation, have to have something. Have to have what? No oxygen. So there are some bacteria that oxygen is actually toxic to. And they have to be in environments where there's little to, if any, little to no oxygen. I'll give you an example of one of these. You guys ever heard of botulism? Yeah. Botulism is a type of what? Food poisoning. And you usually get botulism this way. Let's say that you're in a factory that's making like canned food. Let's just say green beans. You're making canned green beans. Before you put the beans in the can, what do you do at the factory? Well, what do you do to the beans is what I'm saying. What do you do to the beans before you put them in the can? You wash them and you cook them. Hopefully you cook them hot enough to do what? Kill all the germs. But let's say for some reason that didn't happen. Maybe the bacteria had endospores or something. You threw them in the can. Before the can gets sealed, what happens to the can? Or as it's getting sealed, I guess, what happens? They suck out what? Suck out all the air. It's just like a vacuum in there. Well, for a botulism bacteria, that's heaven. Because what do they got? They got plenty of food and no air, which they don't want. So they start eating on the green beans. And what do they give off as they eat on the beans? What will you give off when you eat on things? Waste gases, right? If you ever see a can in a grocery store bulging, that means something's growing in that can. Don't buy it. Don't eat it. Because the toxins made by this bacteria, let's say you bought those beans and you cooked them again, and then you ate them, would you be okay? No. Because the heat would probably kill the bacteria, but they make poison, and the poison is heat resistant. If you were to eat that, you'd probably die. That poison is actually one of the poisons that Saddam Hussein was storing as a chemical weapon in Iraq. It's also the same poison used to do what? Botox. So it makes wrinkles go away, because essentially what does it do to the muscles? It paralyzes them. There are a lot of high-dollar lawyers in places like New York City that are getting their whole face Botoxed, so that their facial muscles, like their expression muscles, they get them all Botoxed. So and some poker players are doing this too. So that nobody can get a read off their face because they can't do anything with their face. They can't smile, can't frown, they can't wrinkle their forehead, because all those muscles have been paralyzed by Botox. Because they don't want to give you any read on them. They don't want you to see anything going on in your face. A lot of lawyers are that way too. Because they never let them see you sweat, kind of thing, if you're a lawyer, right? Speaking of sweat, what do they also do to their sweat glands? They get them Botox, especially like under your arms. If they inject you with Botox, you won't sweat under your arms. It paralyzes the sweat glands, essentially. Really toxic stuff, though. There's another group of bacteria called facultative anaerobes. And those guys we might call the switch hitters of the bacterial world. What can they do? They can do aerobic respiration. They can do anaerobic respiration. They can deal with oxygen. They can deal without having oxygen. They're a little bit of both. This is a picture of just your typical prokaryotic cell. Let me point out some things that we're looking at here. First of all, this one has a capsule, and that's everything in brown. And remember, we said the capsule sort of, oh what? What's the capsule made out of? Mucus. It's like a mucousy coat, mainly there to protect them from what two things? Drying out, desiccating, and immune systems. Just underneath that capsule is the cell wall, and that would be the blue stuff here. And that cell wall is made mostly of peptidoglycan, part protein, part sugar. And then underneath the cell wall, even though it is labeled, it's just hard to see, what's always underneath the cell wall? Membrane, phospholipid bilayer. Same thing you have, just underneath that wall. And then you got the cytoplasm. This would be the one main circular chromosome. This would be one of those little extra pieces of DNA we were talking about, a plasmid. Remember, those are the ones that can do what with? They can swap. They can exchange those during conjugation. And all these little dots in here, those are ribosomes. And remember, ribosomes, what do? They make proteins using directions from right there, or maybe from right there, from the DNA. A couple other things to note. These little hairy-like things on the outside, some of them are called pili. And again, those are the ones that act like Velcro. Some of them might be considered to be cilia, which are involved in what? Helping them move. Some bacteria have larger hair-like things called flagella, which also help them to move. Here we're looking at binary fission. Before this picture ever happened, what would this cell have looked like? Probably would have looked like that. So what happened between here and here? The DNA got replicated, right? It got copied. And notice it's attached to the membrane up top. The cell grows. This copy of the chromosome moves this way. This one moves this way. Until eventually it splits in half. We've got two bacteria that are genetically identical. How long do you think that takes? Sometimes 20 to 30 minutes. If you started with one bacteria and conditions were ideal, by the end of one day, that one bacteria could be several million. Split, 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 splits. Growing how? What's a word for something that's like doubling, tripling? Exponential growth. Growing very, very fast. If we did a growth curve for that, if this was like number of bacteria plotted against time, what shape would the growth curve look like? It'd be like a J. Exponential growth. It just keeps growing faster and faster and faster because 2 to become 4, that's a pretty good jump. But then all of a sudden now you got 4 becoming 8, and then you got 8 becoming 16, and then you got 16 becoming 32, and 32 becoming 64, and I don't know how long I can go with that, so we'll stop there. That's exponential growth. Here's some of those shapes we talked about. Here are cocci bacteria, which are spherical. Now these are in chains. Here's some bacillus, sort of ride, cylinder-like shaped. And then here's one that's sort of spiral-shaped, spirilla. Now, one type of bacteria that we haven't mentioned are these guys, cyanobacteria. For a long time, they were called blue-green what? Blue-green algae. But why was that not a good name for them? Why are they not algae? No. Algae are protists, which means they're what kind of cells? Eukaryotic. These aren't. They don't have a nucleus, they don't have organelles. But they can do what? They can do photosynthesis. So these are photosynthetic bacteria. There are bacteria that can do photosynthesis. And this is one kind, cyanobacteria. And in lots of bodies of water, these are the bottom rung of the food chain. The beginning rung. The most important rung of the food chain. They're photosynthetic bacteria. All right, we'll mention archaea, and then we'll call it a day. When scientists first started talking about archaea as a separate thing, they were finding these bacteria-like places where? Extreme environments. Places that were really hot, really salty, really deep, really dark, really acidic. Extreme kinds of environments. And they thought that that's the only place they were found. But now they've started finding things and doing genetic analysis on them and finding that they're actually everywhere. That they live in like the guts of your guts, for example, your intestinal tract. They're not that uncommon. They're not just found in harsh environments. But the reason they're a different kingdom or a different domain is that genetically, evolutionarily, they're different than what? Regular eubacteria. And like we said before, they're actually kind of in between in terms of evolution, bacteria and eukaryotes. Kind of the missing link between those two groups. A couple of things that make them different from bacteria. One, their cell walls don't have that peptidoglycan stuff that we mentioned earlier. It's made of completely different chemical. And actually, different archaea bacteria have different kinds of cell walls. They all have a cell wall, but it's made of different things. Their ribosomes and their rRNA, their ribosomal RNA, is actually a lot more like ours, like eukaryotic ribosomes, than they are bacterial ribosomes. Like we said, there's somewhere kind of in between bacteria and eukaryotic cells. Why did they get that name archaea? What's archaea mean? It's like archaic. It means ancient. And when they first discovered them, they probably thought that these might have been very similar to the first forms of life. But let's back up to this phylogenetic tree for a minute. Does our phylogenetic tree that I'm pulling up here, does that bear that out? Does that back up the idea that archaea are the most similar things alive now to ancient life? No, actually eubacteria came way before archaea, according to this, right? And again, this is based on what two things? Evolution and genetics. So as they learn more about them, their name maybe not be so accurate. The reason they thought these things were so old is because they thought that the Earth in the beginning was a really nasty place. Really hot, no oxygen, volcanically active, maybe a lot of places were acidic. They didn't think Earth was very hospitable to life. And they realized that some archaea could live in those really nasty conditions. Today, though, our phylogenetic tree wouldn't lead us to believe that these things were first. One other thing I'll mention about them is archaea oftentimes will get grouped into 2 or 3 different groups. One of them is called the methanogens. What do you think that means, a methanogen? These are bacteria that give off methane. Better known to a lot of us as what? Natural gas. Stuff we can burn in your car. To provide energy. These would often be found where, probably? Where can you find a lot of methane being given off, like we're straight into the air? Swamps and stuff like that. And also in the guts of animals, like cows, and you, for example. There are also some archaea called thermoacidophiles. That root phile, P-H-I-L-E, what does that mean? Loving. So these are bacteria that love to eat in the acid. Where do you think those might be found? Yeah, like in Yellowstone, for example. There's a lot of those vents and things in Yellowstone that are really hot and really acidic, down in the vents in the ocean. And then another group, extreme halophiles. What's halo mean? Like halogens. What are halogens? What are halogens in the periodic table? It's group 7, but what does halo mean? Salt. Makes salts. Salt lovers. So places that are really, really salty. Typically, what would salt do to most things? It would kill them by causing them to dehydrate. These things have mechanisms to prevent that. They're just some habitats and shapes of archaea. Places like Yellowstone. Some weird shapes. And then we'll end with this slide. This just compares similarities and differences between the two groups. Some similarities between archaea and eubacteria. They're both prokaryotic. Neither group has an enclosed nucleus or membrane-bound organelles. They both have cell walls. Similar size, usually similar shapes. And they have similar reproductive methods, like binary fission. So these are all things they have in common over here in the left-hand column. And then everything over here in the right-hand column, these are some differences. So the archaea don't have the peptidoglycan that we would find in the cell walls of eubacteria. So archaea have no peptidoglycan. The ribosomes and the rRNA found in archaea are more similar to our ribosomes than they are to eubacteria. And that's why on that phylogenetic tree we saw archaea bacteria is kind of close to eukaryotes. They seem to be genetically related. Also, the eubacteria can make those endospores to protect them against bad environmental conditions. Archaea don't do that. And then finally, so far, scientists haven't been able to discover any archaea that make you sick. Can't find any archaea that are pathogenic for people. That's not to say there aren't any, but so far we don't know of any. All the bacteria that make people sick seem to be the eubacteria. Questions? Think we're at a good stopping point. Let me turn this thing off. I'll leave this on the screen so you guys who are writing can still get it. But I'm going to turn off the recorder.